Hello everybody, this is Jason from STEM Nerds. Thank you so much for being here. In today's video, we're going to be talking about meiosis, and we're going to be talking about how it differs from mitosis, a concept we've already covered. This video is going to be important because in future videos, we're going to be talking about genetics, how traits are passed down from one generation to the next, and it's really important to understand this process before we can understand genetics. All right, so I wanted to start off by showing you a picture of what is known as a karyotype. A karyotype is basically a picture of chromosomes found inside of cells. This is a karyotype of a human cell, and because of that, we have 46 chromosomes. 44 of those are autosomes. 2 of those are going to be our sex chromosomes, either XX if it's a female or XY if it's a male. These chromosomes are arranged based on overall size, and so you can see that the chromosome pair number 1 is the largest, and then as we go to the bottom right-hand side of the screen, chromosome pair number 22, that's the smallest. And of course, we've got homologous chromosomes, one from each parent. What's interesting in this picture is it's actually showing you Down syndrome trisomy 21. If you look at the 21st set of chromosomes, there's supposed to be 2. In this particular picture, there is a third one. All right, so let's get into the process of meiosis. Seems like there's a lot going on here. It's a little bit more complicated than mitosis was in the previous video, but it's actually just the same set of steps repeated 2 times. And so one of the things we have to understand, just like with mitosis, we have a cell cycle. And just like with mitosis, the chromosomes are going to be synthesized or duplicate in the S phase. And so that has already happened. It's not shown here. But by the time we get to meiosis 1, that process has happened. We've already doubled the number of chromosomes. And so here you can see on the far left-hand side, we have interphase. And then in meiosis, unlike mitosis, there is no pro-metaphase. And so we have prophase, metaphase, anaphase, and telophase 1. Now, a couple things to understand here. In prophase 1, we are getting the mitotic spindles forming. The nuclear envelope is going to break down. But you're going to see something a little bit different, and that is we have what are known as homologous pairs that are forming in prophase 1. Homologous pairs are basically sets of sister chromatids. And so now we've got essentially what are pairs of pairs. In mitosis, we had sister chromatids. Here we've got homologous pairs. And those homologous pairs are going to be combined. And then you're going to see they're going to line up at the metaphase plate in metaphase 1. And then the homologous pairs are going to separate in anaphase 1. Now you're going to notice something a little weird, and that is we have the colors are mixed together, the blue and the red. And that is a process called crossover. And so in prophase 1, there is a potential for homologous chromosomes to cross over with one another, and we get a mix of chromosomes from male and female. And so what that means is by the time we get through meiosis 1, we're probably not going to have cells that are genetically identical to the parent cell like we did in mitosis. And so yes, we have some crossover happening in prophase 1. So we have prophase 1, mitotic spindles form, crossover occurs, nuclear envelope breaks down. Metaphase 1, we have the homologous pairs that are going to be pushed towards the metaphase plate. In anaphase 1, it is the homologous pairs that are separating. Then we have telophase where we have 2 cells, and now we have our sister chromatids. Now there's going to be another round of cell division. That is the purpose of meiosis 2. And so you're going to see, once again, we have prophase, metaphase, anaphase, telophase. But here, we're no longer dealing with homologous pairs. Now we're dealing with sister chromatids, similar to what we were dealing with in mitosis. And so just like we talked about earlier, in prophase 2, you've got mitotic spindles forming, nuclear breakdown. In metaphase, now it's the sister chromatids that are going to be pushed to the center. And then in anaphase 2, it is the sister chromatids that are pulled apart into individual chromosomes. Then in telophase 2, we have yet another round of cell division. And so what we started with in meiosis is one cell that had its genetic information doubled. It was diploid. And now we have 4 haploid cells. Haploid is half the number of original chromosomes. And so if our somatic cells, like for us, human beings, we have 46 chromosomes. That is our diploid number. Our haploid number, the number that we find in sperm and eggs, is going to be 23. And so this process is only happening to make sperm and eggs. It's the only place in the human body where we have haploid cells. Now, something else to consider and something that we talked about with mitosis is, let's say you have a cell that has a diploid number of 8. All right, so that's the number of chromosomes that would be in all the somatic cells. If you have a diploid number of 8 in S, we're going to get a doubling of that. We're going to go from 8 to 16. And then when we get to anaphase 1, we're going from 16 back down to 8. And then we get to anaphase 2, we're going from 8 to 4. And so when we started with a cell that had 8 but doubled to 16, we wind up with 4 cells at the end, each with 4 chromosomes. So that's their haploid number. All right, so let's talk about some differences between mitosis and meiosis. First thing is, we already talked about it, in meiosis, we're going to have some crossover occurring. Also in meiosis, we are going to start with diploid cells, but we are going to end with haploid cells, and therefore you're going to have, instead of 2n, you're just going to have n. Once again, when it comes to meiosis, we're probably not going to have genetically identical cells at the end for 3 reasons. One of which we talked about, and that was crossover. The second thing is random mating and random fertilization. And the third thing that we have is something called the law of independent assortment, where those homologous pairs can be flipped at the metaphase plate. As long as each new cell has a full complement of genetic information, they can be in any order. And so because of those 3 things, you're not going to have genetically identical daughter cells when you compare them to the original cell. Also, keep in mind when it comes to meiosis, we're dealing with homologous pairs first in meiosis 1. Those separate in meiosis 1. In meiosis 2, we're dealing with the separation of sister chromatids. We don't really have that in mitosis. We have sister chromatids that separate in anaphase, and then we're back down to chromosomes. So once again, this process of meiosis is happening to produce sex cells, sperm and egg. Our somatic cells, our body cells, are going through mitosis, the process that we talked about in a previous video. In mitosis, what you're getting is hopefully genetic clones. The daughter and the parent cells should look exactly the same, and that's really what we want if we've got cells that are dividing in our organ systems and epithelial cells. But in meiosis, that's not what we want. We don't want exact copies from parent to offspring, and we don't have that. That's why offspring look different than parents. So essentially what we have is mitosis is a form of asexual reproduction, and meiosis is a form of sexual reproduction. All right, everyone, so that is a brief overview of meiosis. As always, thank you so much for being here. In future videos, we're going to be talking about how these traits get passed down from one generation to the next. As always, have a great week.